For millennia, humanity has been captivated by the topic of how humans perceive the physical world through somatic experience. Temperature and pressure activate various kinds of nerves in the skin, which was discovered in the first part of the 20th century. However, until the Nobel Prize-winning findings this year, the identity of the molecular transducers responsible for sensing and converting heat, cold, and touch into nerve impulses in the sensory nervous system remained a mystery. David Julius and Ardem Patapoutian received the Nobel Prize in Physiology or Medicine in 2021 for their discoveries of thermal and mechanical transducers. In the late 1990s, David Julius at the University of California, San Francisco, worked on a quest to find the capsaicin receptor. He used capsaicin, the active component of chili pepper, to identify the sensor in the nerve endings of the skin which respond to heat. Capsaicin was already known to stimulate nerve cells, causing pain feelings, but exactly how this molecule did so remain a mystery. Julius and his colleagues constructed a library of millions of DNA fragments corresponding to genes expressed in sensory neurons that respond to pain, heat, and touch. They hypothesized that the library would contain a DNA fragment encoding the capsaicin reactive protein. Individual genes from this collection were transfected into cultured HEC293 cells, which do not typically respond to capsaicin. After a long search, a single gene was discovered that was able to make cells sensitive to capsaicin. The gene was found to be in code for an ion channel protein TRPV1. Independently of one another, both Julius and Patapoutian used the chemical substance menthol to identify TRPM8, a receptor that was shown to be activated by cold. While the mechanisms of temperature feeling were being discovered, it was still unknown how mechanical stimuli might be translated into our touch and pressure sensations. Ardem Patapoutian was interested in identifying the receptors that are activated by mechanical stimuli. He and his collaborators first identified a cell line that produced detectable electric signal when individual cells were poked with a micropipette. The receptor triggered by mechanical force was thought to be an ion channel, and 72 candidate genes encoding potential receptors were discovered as a following step. To find the gene responsible for mechanosensitivity in the cells examined, these genes were inactivated one by one. After a lot of work, they found the single gene whose silencing makes the cells unresponsive to poking with micropipette. The gene was found to be piezo1, later a similar gene was also identified, piezo2. Further studies identified that piezo1 and 2 are ion channels. They also regulate additional important physiological processes like blood pressure, respiration, and urinary bladder control. The revolutionary discoveries of the TRPV1, TRPM8, and piezo channels have helped us to better understand how heat, cold, and mechanical force are perceived and converted into nerve impulses that allow us to sense and adapt to the environment around us.